All right. Good afternoon, everybody. Hope you guys are doing well. Thank you for joining us today as we move into chapter 6. It's going to be all about the osseous system or osseous tissue. Okay, so this is bone. We're not going to be talking about the names of bones and bony structures. We kind of beat that to death in lab, right? This is mainly going to be looking at the actual internal structure of our bones, the tissue types, the cells, the matrix of what makes our bones and how they work. Um, so before we get started, I want to acknowledge we do have a special visitor today. The Dean of the STEM Division, Dr. Wog, is joining us. I don't know if there's anything else you'd like to say as an introduction, but hello, Dr. Wog.
Hello, everyone. I see you all. Have a wonderful class. I'm looking forward to learning more about bones today.
Yeah. So Dr. Wog is here to observe the class as part of my tenure process, which I don't think I've explained in class at all yet. So I wanted to give a bit of an insight into that. As part of the tenure process to earn tenure here, I have 3 different instructors, either within or outside of my department, the dean of our division, and then 1 student from student leadership that all act as part of my tenure committee. And throughout the quarter, they either will observe my teaching live, such as what Dr. Wog is doing today, or will review my Canvas sites or any kind of material that I've been creating. And really, it's just kind of support for me and help with any of my goals to ensure that I am improving as an instructor and becoming the instructor that I want to be and a part of this college in the way that I want to be. So I really appreciate this part of the process and appreciate Dr. Wog joining us today to give me some feedback on how a lecture heavy class like today's class might go. So a little bit of insight for you, Dr. Wog. Tuesdays are typically quite lecture heavy. So what we're doing today is mainly going to be just talking about the content from the chapter. And then on Thursdays, we usually do some type of activity. And so I will be planning a case study type of activity again for us on this chapter. So you guys can look forward to that on Thursday. And so other announcements before we get into the chapter content, I was able to finish grading all of the skeleton lab exams this morning. So if you haven't noticed, if you go into the grade book, you can see the full breakdown, your percentage from the lecture exam portion, the percentage from the lab exam portion, and then the actual grade, which is contributing to your overall points, is the Unit 1 exam average. And again, that was taken from your percentage on both exams, added them together and divided by 2. And so just the average of that percentage. So when you get a chance, please check that out. I will bring both exams to lab tomorrow. If you want to take the time to review any of them or ask me any questions about your grading, you'll have that opportunity. So beyond that, do you guys have any questions, concerns, or comments that have come up since I saw you yesterday? All righty. Well, cool. Well, then let's talk about osseous tissue and bone structure. Right? So again, this isn't so much the naming of bones like what we've been doing in lab, but really looking at the internal composition of these bones and how they're formed and what they do for the body. So we're going to be looking at bone classification, a lot of the vocabulary that comes with the different cell types in osseous tissue and the different structures of our matrix. We'll look at and compare compact versus spongy bone, our 2 main types of bone that are in each of these other bone categories. The periosteum and endosteum, which are the different segments of our bones. 2 different types of ossification or building of bone. And then the role bone plays as storage and homeostasis for our calcium and phosphorus in our bodies. So our skeletal system as a tissue provides 5 main functions. Mainly, or what the bone does better than anything else is acting as supports. Okay? A majority of the mass of our bone is composed of minerals instead of organic portions, so much less cells and proteins and much more of just these mineral hydroxyapatite crystals that we're going to be talking about more in a bit. Those hydroxyapatite crystals act as not only support in the bone to resist force, but also storage for our minerals. We also have storage of some of our fat in our bones and our yellow bone marrow. The red bone marrow in our blood plays a large role in all of our blood cell production, so red and white blood cells and our platelets. And then our bones act as protection for some of our more vital organs, such as under the thoracic cage and our os coxa, our pelvis, protecting the reproductive organs. And then they provide leverage that allow us to move. Okay? So bones on their own give our body the structure, but then the skeletal muscles that attach to and pull on those bones is what allows us to manipulate our bodies and move through our environment. So just as usual, I'm going to try to do as little as possible of just reading to you the words that are on the slide and try to do focus as much as possible on how to apply this info or what kind of function these different structures provide for us or relate to these functions anyways. So we classify our bones by shape. So if you're asked about what criteria we use to classify bones, it is by shape. And so we can categorize bones into these 6 categories, mainly actually these 4, a majority of the bones that we care about or we'll be talking about, or majority of the bones in the body fit into these 4 categories. That would be irregular bones, short bones, flat bones, and long bones. And then we have a couple others that just don't really fit into those categories or have a category of their own, which would be our sutural or Wormian bones that form those sutures of the skull. And then these sesamoid bones, which were named from their sesame seed-like shape, which is really just the patella. So for the rest of these categories, a majority of the bones we're going to be focusing on and looking at their structure for are going to be our long bones and our flat bones. And just as the name suggests, they are characterized by long bones having a very long shape and flat bones looking very flat, okay? Almost like squamous cells or squamous tissue. Our short bones are all somewhat boxy or cuboidal in their shape. They're generally the same length on all sides. These are like our carpals and tarsals, right, that are all somewhat cuboidal in their shape. And then lastly, we have our irregular bones, which, as the name suggests, they're just different than everything else. And so we're given their own category. And these are mainly our vertebrae. So the long bones are those long bones from our appendicular skeleton. Our flat bones we would find in the skull and face and the clavicles. Okay? So we're going to take a closer look at the structure and function of mainly these 2 in this chapter. And so here's a bit of a closer look at that. So one of the main points to notice on this slide is that we do have a separation of type of bone even further, looking at compact versus spongy bone. So compact bone, as the name suggests, is extremely compact. Right? We don't have much space in between it for organic matter. This is mainly those collagen fibers and hydroxyapatite crystals. Whereas spongy bone forms a sponge-looking structure. It is absolutely not spongy. This is still brittle bone with hydroxyapatite crystals and collagen fibers. But it forms what looks like a sponge you might have in your kitchen or the living sponges that we have in the ocean, right? These overlapping trabeculae that create space. And in that space, you would find mainly red bone marrow or yellow bone marrow, which would be fat. So compact bone is almost always surrounding that spongy bone, creating the periosteum, and then the endosteum, the inner portion, is going to be spongy bone. And so in our flat bones, that it's really like a spongy bone sandwich where compact bone is acting as the bread. So we just have a compact bone on both sides and spongy bone in between. But in our long bones, we really only have spongy bone at each of the poles of this bone, you could say. So our 2 epiphyses. So the epiphysis, we have 2 epiphyses, one on each end of our long bones. This has a majority of that spongy bone structure in it. In between these trabeculae, we would have our red bone marrow. And again, red bone marrow is where a majority of our blood cells are being produced, both red and white, as well as our platelets. We then have the metaphysis, which is kind of what is left over from the cartilage that is replaced during development. But we do get somewhat of a line or a development plate there from leftover cartilage. And then the shaft of our long bones are referred to as the diaphysis. So in all of our long bones, we have one diaphysis in the center, the long portion, the shaft. We have a short metaphysis on each side, so two metaphyses, and then our epiphyses at the very ends, which have that spongy bone in it. And so rather than having spongy bone filling the inner portion of our diaphysis, we would have just marrow, really, much less of those bony trabeculae and a lot more of mainly yellow marrow in these long bones, which again is fat. It's our adipose tissue kind of stored in there. So before we move on into looking at the structure of this tissue further at the cellular level, I'm going to do a quick reflection to chapter 4 and have you guys think about what type of tissue did we say bone was? So what type of tissue is bone?
Dense connective tissue.
Cool. A dense connective tissue. Awesome. And what are the characteristics of connective tissue? And what are some things that all connective tissue have in common? Or could anyone identify anything that you would expect to find in a connective tissue? Constance, I think I saw you say something, but you're muted. You're still muted if you're trying to.
Can you hear me now?
Yes, now we can.
Collagen and maybe fibrous tissues.
Cool. So collagen fiber tissues. Awesome. What else? There were three components we said you could always find in connective tissue that make up the mass of connective tissue. So collagen fibers or other fibers.
Ground substance.
Ground substance should be like the fluid in some of those connective tissues. And then what would be our last portion?
In the cells.
The cells. Okay. So we really have two categories. We have the cells and then the matrix of connective tissue. And that matrix is composed of the ground substance or fluid, which can be more viscous or more liquid depending on the connective tissue, as well as the organic protein fibers. And so since we just identified that bone is a dense connective tissue, we're going to be looking at the different cell types that you would find in bone and osseous tissue. We'll be looking at the composition of the fibers, which in this case is going to be mainly collagen. And then the rest of the matrix is going to be composed of hydroxyapatite crystals. So we have various types of cells, and then that matrix again is collagen and hydroxyapatite crystals. So this long, funky name is just describing the mineral deposits that make up a majority of the mass of our bone. And those minerals are mainly composed of calcium and phosphate. They also do have magnesium in there. And these are what are going to be responsible for resisting any kind of compressive force. So this is what gives bones their ability to hold our weight and allow us to stand up, and any other kind of compressive force that would be pushing us downwards or pushing onto these bones. The collagen, these protein fibers that make up the matrix are going to resist any kind of tensile force or twisting force, so tension. Right? You're going to look at an image in a second here that shows the arrangement of these collagen fibers. And just like how in the dermis of the skin, we had those dermal lines that had parallel networks of these fibers in a particular direction, we have that same kind of structure showing up in our bone that allows it to resist that twisting tension in multiple directions. So protects us from those types of forces and maintains that structure. So starting off with these cells that you would find in our osseous tissue. So we have 4 main cell types to be aware of. We have our osteocytes, our osteoblasts, our osteoprogenitors, and osteoclasts. So nice that they were all given the prefix osteo, referring to bone tissue. And we introduced earlier on in the class this root blast versus this root clast and what they mean. So a blast, any kind of cell type with the term blast in it, is going to be a differentiated stem cell. So osteoblasts are there to act as these stem cells to produce our mature bone cells, which are osteocytes. Osteoclasts kind of do the opposite of osteoblasts. So if osteoblasts are there to make more bone cells, and those bone cells make more bone by producing more collagen, producing more hydroxyapatite crystals, osteoclasts are there to do the opposite. Osteoclasts recycle bone. And so this could be used as a way to provide more calcium and phosphate to the body. So break down this calcium and phosphate storage that we have going on from the hydroxyapatite crystals so that we have more calcium in our blood. But they're also really important for remodeling our bones. So during development, as our bones are being produced and developing and growing in length and size and density, these osteoclasts are going to be activated to essentially recycle any of the structures that were there before, and then allowing space for them to be replaced with more dense matrix and more osteocytes. So these two kind of act together to regulate our storage of those minerals, as well as the density of our bones. Osteoblasts are going to make more bone. Osteoclasts are going to reduce that bone or help remodel it. Osteoprogenitor cells are the, I guess, earlier form of the stem cell that would become an osteoblast. And so these osteoprogenitors are typically all the way on the edge of the endosteum, so the outer portion of what is actually the bone. They will differentiate into multiple osteoblasts. Those osteoblasts are going to start migrating towards the center, so deep into the bone tissue. And as they go, they're going to produce more and more of that collagen and more of those hydroxyapatite crystals, eventually surrounding themselves with that matrix. And that's what and then they will mature into these osteocytes. So you see this osteocyte here has a bunch of these little canals that extend from them. Those are extensions of the membrane of this cell, but a majority of what's surrounding it is that matrix. So all this yellow that you see are those hydroxyapatite crystals and collagen fibers. And so these blasts will just keep producing more and more of that, pushing themselves further into the bone as they are surrounded by the matrix. And then the mature osteocyte is there to maintain that matrix. Okay. And that was a majority of what I wanted to say here. Another key term that you may see come up in other sources, I don't use it very much in this slide deck, but the osteoid, this term is referring to the organic portion of our matrix. So the osteoid is referring to the collagen fibers that are in that matrix. So just a key term to look out for. Okay. And so through this process of creating new matrix and then constantly recycling that matrix with osteoclasts, your entire skeletal system will essentially replace itself in about every 7 years. So a bit slower, takes a bit longer than what we were looking at with our integumentary system, but it is constantly recycling itself and regenerating itself. So if we take a closer look at the actual internal structure of that bone tissue, one single unit of our compact bone, or I guess to also clarify, this is looking at compact bone, is an osteon. Okay, so an osteon is referring to a single unit, and that is this whole circle. If we kind of try to follow this outline that started up here, this full circle is one osteon. So each osteon has a very central here canal, which is where the arteries, veins, and nerves that are going to supply and respond to this tissue are going to travel through. And so that's also characteristic of an osteon. If you see all the layers following the same kind of structure and shape around one central canal, that is all part of one osteon. Throughout this osteon, we have a few different layers of lamellae. These lamellae are essentially circles within circles, and I'll show you in a couple of slides here why we have those lamellae or what function they serve, having them separated that way. But within this osteon, all the way to the central canal, you would just have those osteocytes surrounded by matrix, right? With their canaliculi, these canals, extending the plasma membrane between each osteocyte, allowing them to transfer nutrients through diffusion and through gap junctions. But again, a majority of this matrix, a majority of this mass is going to be those hydroxyapatite crystals. What else here? Another important term that we'll be seeing throughout this chapter is lacunae, or lacuna for a single. A lacuna is essentially the term for the little vessel or hole that an osteocyte is sitting in. So if we go back to this image again, right? This osteocyte surrounded by its matrix is within one lacuna. So think about the term cocoon, right? Having a shell surrounding that organism. This osteocyte is in a cocoon that we call a lacuna. So these lacunae are just those little cavities that osteocytes would be found in. So if we look at an image where all the biological or organic tissue has been removed, you can see all these holes showing up. Those are the lacunae where a mature osteocyte would have lived. You can see our really big central canal, which again is there for our blood vessels and nerves. And yeah, so let's look at those lamellae that I was just talking about. So I'm going to skip ahead a bit here just because I'm talking about those fibers. So the reason we have these different rings within rings, all these concentric lamellae around here, is that we kind of get these three different layers of collagen fibers with alternating orientation. So again, just like with when we were talking about skin, we have those dermal lines which had parallel collagen fibers that resist that twisting force and give our skin some rigidity to itself, or some flexibility anyways. The collagen in our bone is doing the same thing. So having it organized into these alternating directions allows for resistance of tension in all directions. Okay? So if you remember, collagen is stronger than steel when pulled from end to end. So it's really good at resisting directional force that is parallel with it. And so having it in alternating order like this allows it to resist force in both of those directions. And so any kind of twisting and turning that kind of stress we're putting on our bones is going to be resisted by those fibers. So now backing up a bit. So some other terminology, periosteum and endosteum are just referring to the various layers within our bone. So the periosteum, peri referring to the outside, periosteum is this outer layer of bone that we have going on here. And then you would find the osteons within the endosteum, the inner layer. So endo, a prefix meaning in or inside, peri, a prefix meaning next to, actually. So the periosteum is that outer layer, endosteum is the inner layer. And then a bit about spongy bone here. So again, what we were looking at before, this is compact bone. These osteons would be part of that compact bone structure. The spongy bone would be characterized by these trabeculae. So all these extensions that look like their own little network, these are still mainly hydroxyapatite crystals. So we don't have any osteons. And the space that is left behind in between all these holes is going to contain our bone marrow. So again, the spongy bone that is in the epiphyses, the very edges of our long bones, we would mainly have red bone marrow. And then that marrow canal in the center would have yellow bone marrow. And this just helps distribute forces that our bones are receiving back onto the compact bone so that we can still have space within the bone for all this marrow, and we can push that force, distribute the force onto the compact bone on the sides of it. So if you see trabeculae, if you see this structure, when we're talking about bone, that is spongy bone. If you see these rings within rings within rings, that is compact bone. Something to keep in mind. Okay. It's just a closer look at those two different portions of the structure. Again, don't just want to read to you guys. So let's get to something helpful. All right. So just a better look or a better reference for what I've been talking about with resisting force, providing that support in our body. So any kind of compression force, like our body weight, again, pushing some compression onto our bones is going to be resisted by those hydroxyapatite crystals that make up a majority of the mass here. And then any kind of lateral tension or twisting is going to be resisted by those collagen fibers arranged in this alternating pattern of parallel fibers there. So the combination of those two together provides a lot of support, a lot of protection, and allows our bones to stay intact, hopefully, and resist fracturing and breaking when under that stress. So these next few slides, we're going to be looking at how bone is formed, how bone tissue is made. So we'll be looking at endochondral ossification, as well as intermembranous ossification down here. So our two main versions of building bone, essentially. So endochondral ossification is describing how long bones are formed. And all of our bone is going to start out as cartilage, or a different type of dense connective tissue. That cartilage essentially creates the template that the bone cells will then start to break down and replace with more bone, with more of that hydroxyapatite matrix and those collagen fibers. So endochondral ossification starts with this hyaline cartilage model, and we'll talk more about the different types of cartilage as we look at joints in the next chapter and in lab. So you will need to recognize hyaline cartilage later on. So we start with a hyaline cartilage model. Essentially, blood vessels will start to penetrate through this outer membrane, through the periosteum into the endosteum, where they will set up what is going to become our primary ossification center. So let me actually get to this slide. Okay, so we started with this hyaline cartilage model, giving the basic general shape that this bone will become. Our blood vessels penetrate into the endosteum and will act as the supply for the nutrients, the molecules that we need to then build new cells, and the minerals that we will need to deposit into these hydroxyapatite crystals. So the chondrocytes that were here to begin with will continue to enlarge and spread out until they eventually die, and they will be eaten up by osteoclasts, and they will be replaced by the collagen fibers and hydroxyapatite crystals that are being produced by the osteoblasts, until eventually the entire cartilage that was there to begin with is gone. It's all been eaten up, and now there's space. And so now we can fill the epiphysis with marrow, and then as we get it, the diaphysis with marrow, and these blood vessels will continue to move on and penetrate into each of our epiphyses. So after this marrow cavity has been formed, and we have our compact bone structure on the outer regions of our diaphysis, those blood vessels are going to continue to penetrate into the epiphyses, where we now get secondary ossification centers. So that primary ossification center was in the very center of the bone in the diaphysis, and now each epiphysis will form a secondary ossification center. And if you haven't caught on, ossification is just the process of making new bone. So these secondary ossification centers will act in the same way. Osteoclasts will start to help break down all the chondrocytes that were enlarging and dying, and then we can start replacing it with bone from the osteoblasts, replace it with hydroxyapatite crystals and collagen fibers. And so in this way, we have now developed a bone. Okay, so this would be during fetal development. After birth, and these bones need to lengthen and increase in diameter, okay, we have two different types of growth within the bone to provide those two different types of growth. So we have one type of growth that gives length to these long bones, and one type of growth that gives width or girth to those long bones, density to those long bones. I'll come back to this in a second. Just because I talked about the growth, I want to make sure I touch on it. So in long bones, after development, which was what that endochondral ossification was, that was development. We then have interstitial growth and apositional growth to increase the length and increase the diameter of these bones. So interstitial growth is new bone tissue being created within the diaphysis that is going to push the epiphyses away from each other. So as the diaphysis is growing and growing, right, both sides ultimately get pushed away, thus lengthening the bone. So interstitial, because it's in between our two poles, interstitial between, pushing those two poles away from each other, pushing the epiphyses away from each other. Apositional growth is there to help increase the diameter of the bone or the thickness of the bone. So apositional growth is going to be a lot of that bone recycling rather than just new deposition of bone like interstitial growth was. So with apositional growth, osteoclasts are breaking down some of the older bone so that there's space to be replaced with new and more osteoblasts, which over time will increase the periosteum, the outer portion, the compact bone on the outer portion, thus giving this bone a wider width, a wider diameter. So again, interstitial is to grow length, apositional is to grow the diameter and density or strength of that bone. Okay, so now backing up a little bit. Something I didn't touch on, the cartilage that is kind of separating the diaphysis and the epiphyses, which is what we call the metaphysis here. That cartilage stays behind for a lot longer than the cartilage that was in these two areas. So the cartilage that was in this metaphysis is what ends up becoming the growth plate, that cartilage growth plate. And if you take an X-ray of a child's appendages or anywhere where there would be bones connecting, you can really see these gaps that sometimes look like broken bone or just look like wide open spaces in between those bones. And that's just because we don't visualize cartilage under an X-ray in the same way that we can visualize bone. So these spaces aren't completely empty the way they seem to be. There is cartilage there that over time is going to be broken down and replaced with bone tissue, with all the structures we've just been talking about. So comparing this child's hand to an adult hand, right, all those bones seem to almost fully connect. Of course, there is space for some cartilage and fluid that makes up the joints between these bones, but much less space than what it looks like there is here. Okay. I realized I've allowed myself to get a bit carried away without checking in with you guys since the reflection part. So how are things going so far? Are there any concepts or ideas that I could explain further or you would like me to touch on again?
I guess just the hydroxyapatite crystals is pretty cool. I was just Googling that real quick as a form of remineralization for teeth. I thought that was interesting. I'm surprised it's not more mainstream.
Yeah, I don't know much about teeth as their own specific bone, but I was really interested. The last time I went to the dentist, they pulled up my x-rays and I could see all the labels with the different types and got to ask them questions for a bit there. And James, you're interested in going into dentistry, right? So if you are going to do more research with this and learn more about it, please share with me. I do want to include more about teeth in here, even though it's not really addressed in the text for this chapter. But it does have slightly different structure to it, or at least specificity to how our teeth are managed and made. And it is cool, right? The strongest bone in your body is in your teeth, right? Or the strongest tissue in your body is in your teeth. The enamel, right? Strongest, most compact version of our bone. Okay. So this was all part of growth and creation of our long bones, right? That was endochondral ossification, and then the growth of these long bones comes from interstitial growth and apositional growth. But we also have intramembranous ossification, which is what forms dermal or flat bones. And dermal bone, that name is referring to the fact that the growth or the beginning of this bone is actually happening in the deeper dermis, so from our skin. That's the dermal bones. So with our endochondral ossification within the chondrocytes, so within our cartilage, we had cartilage making a model that was then replaced with bone. For intramembranous ossification, we do not have a cartilaginous model to begin with. Instead, we have a whole bunch of these mesenchymal cells, and I guess I'll wait to dive into that until we get to the slides that look at it more deeply. But again, this is originating in the deep layers of the dermis, so part of our skin. Again, this is the creation of bone, so it's happening during fetal development. And these are characterized by these spicules that eventually form that spongy structure. So let's take a look at what that looks like. So I know this image ended up a bit fuzzy on my end, but I'm looking at it on quite a big screen, so it's not so bad. Are any of these words, or are these words visible to you guys on this figure, or is this too stretched?
Visible.
It's pretty clear.
Okay, cool. Cool. All right. So rather than having a cartilage model that is then going to be stretched and enlarged until those cartilage cells die and get recycled, creating a space, instead we have a whole bunch of these stem cells, these mesenchymal cells that end up filling this space deep to the dermis. And we begin forming some osteoblasts that create essentially different segments of bone that are all going to end up connecting with each other. Okay, so that's kind of what we see beginning to happen here. We have osteoblasts, or these mesenchymal cells are differentiating into osteoblasts. These osteoblasts start depositing that collagen and hydroxyapatite crystals surrounding themselves in a matrix. And so we start forming these ossification centers all throughout this little tissue. Blood vessels are going to start to penetrate through this and pop up. And so those will become little canals. And as the matrix is getting full more and more, the shape of these little pockets of bone start to form what we call these spicules. So these ossification centers start to form these little spicules, or these little extensions almost of the plasma membrane, or if this was a cell of the plasma membrane, but it's pushing the entire tissue as it's being filled with collagen in those crystals. And so that essentially just continues to happen as the spicules connect with each other, and we start getting more densely compacted bone, leaving space for those blood vessels that are now trapped in this matrix, that have their own space to travel through, until eventually we have completely filled that space with bone, with those crystals and collagen fibers, and we have osteocytes trapped in their little lacunae, right? And so all of this is going to be recycled and remodeled with osteoclasts and osteoblasts until they get their mature shape and structure, which is what you see here. So essentially, early on in bone development, we're just pumping it full of those crystals and collagen fibers to start giving it the shape that it's going to have. Osteoclasts will then come and break some of that down, and osteoblasts will replace it with what is going to be its mature function. So when we're talking about remodeling the bone or recycling the bone, it's just trying to get it towards that more mature structure that performs the job it's supposed to have with those parallel collagen fibers, or in spongy bone with those trabeculae, and getting away from this more flat or random deposition of collagen fibers and minerals. So that is intramembranous ossification. Again, that forms a lot of our flat bones. So for example, the bones of the skull, the mandible, our clavicles, those are all flat bones. And our endochondral ossification, again, was for long bones. Okay. And so the last function or concept that we'll address about bone tissue here is bone acting as our mineral storage, and the role that it plays in homeostasis of these minerals. Okay? So majority of that hydroxyapatite crystal, or backing up even further, a majority of the mass of bone, again, is the inorganic portion of the matrix. It is those hydroxyapatite crystals. Okay? So 2/3 of the bone's mass is inorganic. It is these minerals arranged in that structure. So only 1/3 is actually composed of organic portions. So those would be proteins, carbohydrates, lipids, and nucleic acids. And a majority of that organic component is really just the collagen. So a very, very small portion of what is making up the mass of our bone is the living, functioning cells. Most of the mass is there to provide that support. Okay? And so those hydroxyapatite crystals themselves are made up of various minerals within them, mainly calcium and phosphate. And as we've talked about a few times in this class already, calcium is going to be really important when we start looking at signal transduction And especially when we look at muscles and allowing for our muscles to actually contract, allowing for those little myosin heads to attach to the actin filaments and pull those actin filaments, which is what makes our bones or our muscles shorten and pulls our bones towards each other. That's going to heavily rely on calcium. Phosphate, of course, is a very important component of ATP, as well as being an important component of our nucleic acids that make up our DNA and RNA. And so having somewhere where we can store those extra minerals so that we have them for when we need them, and then regulatory processes that are responsible for determining how much is stored and then how much is being released into our bloodstream is going to be really important for the function of all of our cells, for our entire body. And so not only is the composition of bone mainly minerals, but a majority of those minerals that are in our body are in your bone. 99% of the calcium in your body is stored in your bone. 88% of the phosphate is stored in your bone. Okay? So very important as far as participating in that homeostasis of these minerals. And so we produce a few different hormones that are going to play a role in how much calcium is available to your body. Again, to help with that signal transduction, to help with muscle contraction, to help with providing energy, right, ATP. And so I took this table from the text and added some little arrows to it. And these arrows are indicating the effect that each of these hormones has on the overall blood calcium levels. So this does not directly correlate with what effect they would have on necessarily bone density, because some of these are related to absorption of calcium and phosphate from digestion, such as calcitriol, which is vitamin D. Right? This increases how much calcium and phosphate is in the blood, but doesn't necessarily affect our bone density, or isn't decreasing bone density to do it. Whereas something like parathyroid hormone, or PTH, is also going to increase the amount of calcium in the blood, but it's doing that by breaking down our bone, right? By stimulating osteoclasts to elevate the amounts of bone that's being broken down and recycled, thus decreasing bone density, so taking calcium out of our bone and phosphate out of our bone to get it into the blood. So just things to keep in mind, right? So we do have multiple hormones to look at here, and a couple figures that exemplify their effects pretty well. But calcitriol, again, is vitamin D. This is the active form of vitamin D. So after you've been exposed to the sun or get vitamin D through your diet, that inactive or immature version of vitamin D needs to eventually be activated into calcitriol through the kidneys. But the active form of vitamin D is going to help promote calcium and phosphate absorption through our digestive tract. So this is really helpful in activating our genes that are responsible for creating the proteins that will transport calcium and phosphate, or even for creating the proteins that are going to open protein channels to allow those minerals to come into the cell from the lumen of our intestines. We have growth hormone, which is from the pituitary gland, which stimulates the deposition of calcium and phosphate into our bone. So it's going to stimulate these osteoblasts, so making more of that bone matrix. So it's going to use up the calcium and phosphate that we have available in order to store it into the bone. Thyroxine from the thyroid glands works hand in hand with growth hormone to stimulate a similar response with our osteoblasts. Our sex hormones also stimulate osteoblasts to help with growth. We already talked about parathyroid hormone. And so lastly, calcitonin, rather than just stimulating the osteoblasts, which are going to create more matrix, they actually actively inhibit the osteoclasts, which would be breaking down that bone matrix. They also promote calcium loss at the kidneys and reduce overall those calcium ion concentrations. So let's take a closer look at that. So we've got two figures looking at factors that increase blood calcium and looking at factors that decrease blood calcium levels. And so basically the threshold that is going to stimulate this response, stimulate this release of hormones, is any ion concentration below 8.5 milligrams per deciliter, or any ion concentration above 11 milligrams per deciliter. So if we have less than 8.5, our organs are going to release the hormones that are trying to stimulate more calcium and phosphate into our blood to try to get that ion concentration higher. And if that ion concentration exceeds 11, milligrams per deciliter, we're going to release hormones that try to remove it from our blood. So essentially the sweet spot is right in between. We want these ion concentrations to kind of stay between 8.5 to 11. And these other regulatory messengers, those hormones, are there to try to keep it that way. So if our calcium ion concentrations are getting too low, we want to make sure we have more calcium available for signal transduction, for muscle contraction, etc. Our parathyroid hormone is going to work alongside vitamin D to help increase absorption, to tell our kidneys to retain as much calcium as possible, so stop peeing it out, filtering it out and peeing it, and can also stimulate our osteoclasts to activate, which is going to break down some of our bone to release more calcium and phosphate into the bloodstream. So all three of these reactions are going to help increase that concentration level. So getting calcium from our diet from our intestinal lumen and into our cells or into our bloodstream, keeping whatever calcium is already there by inhibiting our kidneys filtering it out and excreting it with their urine, and then activating or stimulating these osteoclasts, which will break down some of our bone, releasing that calcium and that phosphate to go back into the bloodstream. So again, hopefully these will all increase the concentration available in our blood. So on the opposite end of the spectrum, when our blood concentrations are getting too high, our parathyroid hormone and calcitriol levels are going to decrease, or we'll try to inhibit how much of those we are producing, thus decreasing how much calcium we are actively absorbing from our diet. Calcitonin is going to stimulate the filtration of calcium in our kidneys. Thus, we will be excreting more of it in our urine. And calcitonin is also going to act directly on our bones by inhibiting the activity of osteoclasts, thus allowing osteoblasts to continue taking calcium and phosphate out of the blood and start storing it into bone. And that is essentially everything from this chapter that I wanted to make sure we address. So are there any parts of this second half that you guys would like me to touch back on, or really from any of this chapter that we've talked about? So are there any ideas you guys would like me to touch back on, go more in depth, or that you have questions about?
For increased osteoclast activity, is there a region of the body where that would increase more to release this calcium?
That's a
Like,
good
is it
question.
more in long bones, or like does it depend on like what area is lacking and like just bones in that region?
So at this moment, I don't know the answer to that question, but it's a very good question, and I would like to know. So I'm going to try to make a note of that somewhere. So I will have that answer for you at next class for sure.
Thank you.
Is it possible for you to reiterate the main points that we should know about the endocondyle ossification?
Yeah. So the number one main point is there is a cartilage matrix that will be replaced with bone, right? That is the major difference between these two types of developmental growth of bone. Endochondral, meaning inside the cartilage, it's going to start with a cartilage matrix. And so this is what makes our long bones. The formation of the diaphysis essentially happens first. Okay? As the blood vessels penetrate into here, we'll then start breaking down, creating this hole, that marrow cavity, as the chondrocytes grow and die, and we replace them. After the diaphysis, the space within the diaphysis has essentially been formed, it then penetrates into each of the epiphyses, so our two poles, where we will continue that same cycle. So chondrocytes grow and die, and they are replaced with the minerals from our bone, or just with open space, which is going to be bone marrow. The main point is that it has a cartilage model, and that this makes long bones, I'd say. And so beyond that, it's just getting into specifics, that the diaphysis forms first, creates the marrow cavity, and then penetrates into the epiphyses. But there's two quizzes for this week for chapter 6 content. Both of them each touch on endochondral ossification and the different types of growth, as well as the difference with intermembranous ossification. So you do get a good bit of practice with answering questions about ossification as well as mineral storage, since those are quite specific.
Thank you.
One other fact that I don't think I mentioned and want to make sure I make note of it, something unique about osteoclasts, they are much more like immune cells than they are like the rest of our bone cells. So these osteoprogenitors do not differentiate into osteoclasts. These stem cells become osteoblasts, and those osteoblasts become osteocytes. But osteoclasts are much more like immune cells or blood cells. They're actually formed in the red bone marrow, and they're formed by fusion of multiple stem cells together from that bone marrow. And so that makes this really large multinucleate cell that's very much like a macrophage, right? That immune system blood cell that's supposed to destroy invaders. This osteoclast is much more similar to a macrophage than it is to an osteoblast or any of the other cells here. And again, it is multinucleate. There are multiple nuclei in these osteoclasts. So I think the only other cell type we've introduced so far that is multinucleate is skeletal muscle cells, right? So we just had those two so far that have that unique distinction to them. Any other questions for today, you guys? All right. So as I mentioned at the beginning of class, I will bring both of the exams that you guys have completed with me to lab tomorrow. So if you're curious and want to look at them, or if you want to make any arguments for points like we did with the lecture exam, I'm more than open to that. So there will be some time for that. Tomorrow in lab, we're mainly going to be focusing on the joints of the rotator cuff or the shoulder, and introducing different types of movements that happen at various joints. So that's going to be our starting point as we move into muscles, which will be our unit 2 focus for lab. Is all about muscles and the movements that they do. And then on Thursday in class, I'll have an activity likely very similar to the case study type of activity we did with tissues for us to work on Thursday. So if you don't have any other questions, concerns, or comments, you guys are free to take this time back and enjoy the rest of your day. And I will see you tomorrow for muscles.
Thank you.